First disease we're going to discuss is malaria. Okay, malaria is one of the oldest diseases that has afflicted humans. It's been around. It has had major impacts on our history. It ranks right up there with the Black Plague, with tuberculosis, influenza, not only for the numbers of people afflicted, but the number of bodies associated with this disease. Even today in our modern world, with our modern medicine, they calculate that about 40% of the world's population every year is exposed or threatened by malaria. Malaria has a certain progression to the disease. Once you're infected via the mosquito bite, there is a incubation period. Incubation period that's going to last 10 to 16 days. So, week and a half, 2 weeks after you are bit, and you're not going to know which of the mosquito bites is going to be responsible on which day. You're going to start coming down with the first symptoms. You look at the list here of symptoms: malaise, fatigue, aches and pains and nausea, chills, fever, sweating. The severity, as we're going to see over the next few slides, will be dependent upon which of the different species that are known to cause malaria you get infected with. The interval, 48 to 72 hours. We're going to see as the protozoa infects cells, your symptoms start to decrease for a few days, and then those protozoa cause the red blood cells to lyse as they spread to new blood cells. And they continue on, over and over. So when you see or you read about the clinical presentation of malaria, it's not just this one constant, I feel sick for a week or so and then start to recover. No, it's this cyclic 2 or 3 days of really bad, then a couple of days of feeling a little bit better, then back to the bad, then back to a little bit better, back to the bad, and it just keeps going, cycling back and forth. And it's all dependent, like I said, on which of the different species you get infected with. What we do know is many of the symptoms are going to be due to the lysing of red blood cells. Now, it's not all the red blood cells in your body all at once, because that would kill you. But it's a nice percentage of them. I mean, we're talking, 1, 2, 3%, which doesn't sound like a lot, but spread throughout your body, all of these lysing within a few minutes, a few hours of each other will start to cause your body to react. What you actually end up with is an inflammatory response body-wide to the contents of the red blood cells. Things that your immune system should never see. The interior contents of one of your cells will now start triggering immunoresponses. On the more severe cases, you can end with what is cerebral malaria. Plasmodium falciparum being the species. Turns out falciparum is actually the most severe of the species, the different species. What we find here is that during the cycling, getting into red blood cells, getting out of red blood cells, back and forth, will cause some of the red blood cells that have lice to start sticking to hold red blood cells. So you start forming clots. The clots don't completely block the blood flow, but they will start to diminish blood flow. And in some severe cases that I remember reading previously, years ago, will not only start causing bleeding into the brain, which allows for falciparum to get into bypassing that blood-brain barrier and getting into the central nervous system. Most cases, though, it's going to lead to decreased blood flow into the meninges, meaning decreased blood flow into the brain, meaning decreased oxygenation, which is going to lead to coma, death. Now, on the scale of the different species, Plasmodium falciparum being the worst, below that you have Vivax, Ovale, and I do believe when it comes to clinical settings, clinical presentation, it's actually Plasmodium malariae that is the least deadly. But don't quote me on that. So when you go through and you start looking at the different Plasmodium species that can infect humans, malariae, Vivax, Knowlesi, Ovale, falciparum, each has a different regions around the world. Now, they're not distinct regions. There may be overlap. But there have been reports that by seeing which species a person was infected with, you can narrow down which regions of the world they have been in. What we know when we look at the life cycle of Plasmodium, we see that there are two phases, an asexual and a sexual phase. Asexual means that binary fission. One Plasmodium splits becomes two. Two split become four, and so on. Sexual phase means that they actually take on the role of gamete. So a quote-unquote male and a quote-unquote female will fuse together, mix genetic contents, and then divide. By mixing genetic content, the resulting offspring, while still the same species, will have a greater chance of being genetically unique. Kind of the same vein of how you're a little like mom, a little like dad, but still genetically your own. When it comes to parasites, it is a rule that whichever host the sexual reproduction takes place in, that is the definitive host. In our hubris, we humans like to think that we are always the definitive host. And here with Plasmodium, that's not the case. You get bit by a mosquito. Turns out, when that mosquito goes in for the bite or its blood meal, it pushes its proboscis through the outer epidermis into the underlying dermal layers, going for the subcutaneous dermal layers where the capillary beds are. Once the proboscis is able to insert into a capillary, the mosquito will spit out or regurgitate saliva. In the saliva are anticoagulant proteins. The anticoagulant proteins minimizes the chance of your blood clotting. No blood clot, the blood keeps flowing. That's why when you look at a mosquito bite, sometimes you can see a small little microscopic drop of blood, little bitty drop, even after the mosquito has left, because that anticoagulant is still allowing the blood to flow out. Well, here's the thing. When the mosquito spits out the saliva, splits out whatever, not only are those anticoagulant proteins entering into your body, but there's also the parasite, Plasmodium. Plasmodium, as you see here, looking at this figure from the textbook, the Plasmodium enters into the bloodstream and will exit the bloodstream in your liver. So the first step is infecting hepatocytes. One goes in, hundreds, thousands come out. And it just keeps going, spreading liver cell to liver cell to liver cell. After a given amount of time, there's been a high enough population increase. They begin to leave the liver, entering into the bloodstream, and then infecting the red blood cells. Here, as it's going into the red blood cells, you will see a maturation process. One goes in, dozens come out, spread to neighboring red blood cells, and they go through this cycling as they begin to mature. All of this increase in numbers in our body is via binary fission, asexual. These will reach a certain point, these sporocytes, until they will then switch on certain genes that will cause them to become gametes. Gametes that will be taken up during the next blood meal by another mosquito. They go into the mosquito. Once they're in the foregut, hindgut area, they start fusing together to start forming zygotes, which will go through cellular division, and you get new Plasmodium. So we like to always think of the mosquitoes as the vector spreading malaria person to person to person, when in reality, we humans are actually the biological vector spreading Plasmodium mosquito to mosquito. So you look here on this slide, this pretty much covers a lot of what I've talked about, so I'm not going to go through it too much, so you can review this and read the chapter. Turns out when it comes to mosquitoes, it's the female that goes in for the blood meals. Okay, so it's the female Anopheles mosquito that we worry the most about. She goes in for her blood meals, helps with the making of the eggs and all that stuff, keeps her nutrition levels high. So if we can figure out a way to interfere with either the blood meal or with the reproduction, we can somehow start to control this. World Health Organization is one of the organizations that's tracking malaria worldwide. 200 million new cases every year. Okay, that's not 200 million people sick every year. That's 200 new cases. Okay, 200 for 2019, 200 million more 2020, which means 400 total, minus those that succumbed to malaria. They think anywhere from 0.5 to 1 million people die annually. Now, in some places, this is actually a childhood disease, okay? Childhood disease means you're going to be exposed in childhood, And this is going to be one of those steps that you have to be able to survive. If you can survive it in early childhood, adolescence, there's a pretty good chance you can survive through adulthood. Here in the US, we actually do have a problem with malaria. It's not endemic to our area. What we see in the clinical standpoint, in the clinics, in the ER, when patients present with symptoms that scream out malaria, more often than not, these are immigrants from areas where it is endemic, or American tourists who went, visited, got bit, came home within that 10 to 16 day incubation period. When you look at the map here and you start looking at the areas shown in yellow, around the equator and between the two tropics. So this is a tropical. A lot of times students will ask me, well, how come if it's tropics, why do we not see it so much, as an issue, North Africa, southern China? Well, thing to keep in mind, North Africa, southern China, and, northern Mexico, desert. Okay? North Africa, Sahara Desert. Southern China, Mongolian Desert. Northern Mexico, desert. Okay? Deserts don't have a lot of water, and mosquitoes need water for their larval stages. So that's why you see, when you look in areas, northern South America, sub-Saharan Africa, basically the Far East. These are going to be areas that have lots of monsoon rain, lots of water. Their yearly rainfall can be measured in meters in some places. When it comes to diagnosing malaria, not only do you go by the symptoms, the cyclic nature of the symptoms, but you do a blood smear. Take a few drops of the blood, smear it on a microscope slide, fix the red blood cells, and then stain them. You look here. These red blood cells. Notice how each of these red blood cells, as you see right here, have this little ring shape in them. Yeah, that ring shape is actually a diagnostic tool for malaria. You get a really good parasitologist in your associated with your ID lab. They can look at that, not only look at those rings, tell you what stage of development the sporozoites are in, but for some of them, they can actually give you a pretty good indication of which species. When it comes to prevention, there have been lots of things tried. Draining of swamps, getting rid of standing water, spraying DDT and other, anti-bug sprays. They have had some mixed results with those, but one of the greatest advances in Africa, in the Asian countries, Southeast Asian countries, in diminishing the number of cases year to year of malaria, has been shown what's right there to the right in that picture. Mosquito nets. Okay, yes, you go outside throughout the day, there's a chance you can get bitten by mosquitoes. Okay, we all know that. But we also know the greater probability of you getting bitten is going to be dusk throughout throughout the night. That seems to be when you get the greater numbers of mosquitoes out and about. So what? A few decades ago, can't remember, late 80s, early 90s, I believe, they started distributing mosquito nets. I mean, we've had them around for a long, long time, okay? They've been around for over a century or so, okay? But these were more of a thing that only people with money could afford. But as, the manufacturing processes, throughout the Industrial Revolution, throughout the 20th century, became cheaper and cheaper to make these fine nets. Got to the point where they were so cheap, we could afford to just give them away. So by distributing these throughout, sub-Saharan Africa, throughout Southeast Asia, sleep under these, minimize your chance of getting bit by mosquito, minimize your chances of contracting malaria. We've been working towards a vaccine, okay? The nets are great when you're under them, but you're still going to be out and about at some point in time during your day, and there's still a chance. So, they're looking at vaccines. Now, it's hard to come up with a vaccine for malaria. Okay, malaria, Plasmodium, has slight abilities to not evade the immune response. It's able to slightly alter the proteins and sugars on its surface during the different stages of its development, so that by the time your immune systems learn something, not you is there, well, it's switched up its protein, switched up its sugars, and your immune system has to relearn. So they're trying to develop a vaccine that will compensate for this. They've chosen Plasmodium falciparum as the first vaccine candidate because falciparum, as I said earlier in this lecture, is the deadliest. So they're developing vaccines. RTS is one of them specifically. They are clinical tests. They've shown that it works great in children. Get it in children, hopefully you get a lifelong immunoresponse, that immunological memory. Beginning a couple of years ago, 2018, they have started pilot vaccination programs in Africa. For those who have already been infected, vaccines only work if you haven't been infected yet. Once you are infected, you have malaria. There are clinical cases where malaria is going to be something you have for life. Okay, it becomes a chronic hepatic infection. Okay, it's a low-level infection. It stays in your liver, and during certain periods of stress or whatever, years later, you may get the reemergence of malaria. So what we have found is it's best that once people start anti-malarial drugs, just keep them on it. Quinine was the first. Okay. Quinine, remember that's the old gin and tonic story, that urban myth I told you about? Well, we now have chloroquine, which is less toxic. Quinine can have some levels of liver toxicity associated with, so we switch over to chloroquine. Chloroquine works good. It does its job, but the problem we have noticed is that there are strains of the different Plasmodium species that are becoming resistant. How do they become resistant? Well, maybe there's an interruption for a few days to the chloroquine. They ran out. It's going to be a while before they can get the next. Well, that allows for reemergence for whatever, sporozoites in the bloodstream or in the liver that weren't killed by the chloroquine. They come out, they have a slight resistance, they spread, you start taking it again, you maintain it for a few months, a few years, and then you interrupt again, they come out again, and it's a back and forth. We've talked about this whole thing, the 99.99% effective, blah, 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 still leaves you 1 in a million. That's going to be a little more resistant every time. So we're starting to find resistant strains popping up around the world across the different species. Falciparum, Malariae, Vivax, whatever. We're now looking at artemisinin and plant compounds. Turns out Mother Nature's got a lot out there we're still learning. We're trying to use this as part of a cocktail. Okay, artemisinin by itself isn't perfect, but we're starting to see that as a cocktail with other chloroquine-based compounds, chloroquine derivatives, that, well, we may have something that's going to show, greater chance of, or a better chance of exhibiting non-resistant strains for much longer. We'll see how this goes.